In this video, we will discuss about the results that will show that FAS ligands or FAS signalings blocks the CAR T cells apoptosis. So, in order to check the effect of FAS on the CAR T cells, they develop two different kind of CAR Ts. One that have the intercellular domains that can trigger apoptosis, and the other things that does not have FAS or that does not trigger apoptosis once it interact with the FAS ligand. And they check these cells at three different conditions. First, the untransduced cells, T cells, and they check the signals before the cells come in contact with FASL ligand, and 4 hours after the cells come in contact with FASL ligand. And they can see that the untransduced CAR or the T cells which are not been transduced with CAR, they see around 46% rise in the active caspase 37 signaling or apoptosis. And when they use the transduced CAR T cells, that is the normal CAR T cells with the intercellular domains, they see around 78.9% of the apoptosis. And when they transduce this CAR T cells along with this FAS ligand, or where the intercellular domains is not there, they do not see any apoptosis. Only 10% of the apoptosis is there. So this indicates that these FASL ligands will trigger apoptosis, and its inhibitions will prevent the apoptosis. And then they presented here the SPICE analysis. Here we can see that the T cells, the TNF signaling, when the T cells alone are there, and in the case where they are transduced with CAR T, that is this normal CAR T, they can see some TNF signalings, and in the normal CAR T with this FAS ligands FAS signalings has been deleted, they see a slightly lower ECF. And when they use this PMA, which is used to activate the T cells, they see a high amount of TNF signalings, INF gamma signalings, and interleukin 2 formations. When they check it for the T cells with the K562 CD19 cells, that is used to activate T cells, only in the case of CAR T cells without FASL ligands decrease, they see TNF signalings and INF gamma and some amount of interleukin 2. And when they use the FASL ligands, or when they remove these FASL ligands along with this CAR T transductions, they see a slightly more amount of interleukin 2, and this could be because of less apoptosis. And these things they have also did the cytotolic assay, where they can see that in the case of where the cells are M-cherry fluorescence, so they see the M-cherry fluorescence when the cells are transduced with CAR T or CAR T that have with this FASL signaling is reduced. And what they are able to see that M-cherry signals is quite high in the case of untransduced cells, whereas when the cells are transduced with the CAR T based when the T cells are transduced with CAR, they see a decrease in the M-cherry signals of the cancer cells, and which are fluorescence with this M-cherry. And when they use the ECF, that is the CAR T with this FASL ligands is removed, they further see a decrease in the signals. And they can see the p-values are shown here between the untransduced and the EC, and untransduced and ECF decrease is significant, but EC and ECF is non-significant. And they check the activations of the T cells at different conditions. They can see that the T, and with the introductions of this PMA, when the PMA is not there, they see less activations. When the PMA is there, they see high activations. And these things they have quantified the panel F, where we can see that when the PMA is there, the all threes are activated to similar extents. That can be seen here. One thing that we need to understand, that when the PMA is not there, even then the CAR T cells are activated in both in the EC case and ECF case. That is, the CAR T are transduced with intracellular domain, and when the CAR T are transduced without delta FAS signalings. And thereafter they check the HLA, which is the proteins that are expressed on the cytotoxic T cells, and this anti-HLA can directly detect those peptides, whereas flu and EIK3CA can detect those peptides in the viral parts. So they can see that these HLAs are present in the viral particles, but they are not present in the T cell surface after day 2. And these things they have further quantified, or reproducible results are shown in the panel H, where we can see that the cell surface expression of HLA goes down, which is the marker for cytotoxic T cells, whereas in the case of virus titer, they are still present at the similar amount even after 3 days. So this altogether indicates that when the FASL is present, when the FASL signaling is present, it can trigger apoptosis in the T cells that are transduced with CAR T, and its cytotoxic effect is been shown, or effect of T cell effectors effect has been shown. This cytotoxic effect through the HLA protein expression on the cell surface has been shown in the panel G and H, which shows that its cytotoxic effect goes down with time. And this is in the case where the T cells are transduced with this LNGR or EGFR receptors. So this indicates that its cytotoxic effect will also goes down with time. That's all for this video. I hope you understood that how this shows that FASS will cause apoptosis. One of the reasons could be this intracellular activations of this CAR T that has been transduced, as with time those cells triggers apoptosis, and their characteristics of cytotoxic effect, which is tested by this HLA protein surface expression, is also goes down, whereas in the virals titers peptides, they are still present. So I hope you understood to some extent this, how the FASL signaling is related to the CAR T cell apoptosis. If you need to understand more about their uh, relationships, or how they did their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.